Okay, so welcome back to this next video in which we are discussing the hemostatic pathway, so how we stop hemorrhage from a cut that we have made in the side of an arteriole. Okay, so we've discussed so far that what happens is when you cut the side of an arteriole, what will happen is that will expose von Willebrand factor on the in the membranes of these endothelial cells to the platelets within the blood, okay? And then the platelets will bind to the von Willebrand factor via glycoprotein 1b95, okay? And this is called platelet adhesion. Then what's also going to happen is that platelets are going to come across collagen and tissue factor, and the collagen and tissue factor are going to lead to them activating. And when they activate, they start releasing ADP, adenosine diphosphate, and thromboxane A2. Now, ADP triggers off a chain reaction where it binds to ADP receptors, and of course in the usual physiology you would not have clopidogrel floating around bound to the ADP receptors. So the ADP binds to the ADP receptors, activates more platelets, so you set off this chain reaction where you're just getting more and more thromboxane A2 overall released. And thromboxane A2 has two effects. Firstly, it's going to cause the vasoconstriction of the arteriole to limit the amount of blood that is getting to this portion where you have the cut. And secondly, it's also going to bind to the glycoprotein 2b/2a, and this is going to lead to the activation of this glycoprotein 2b/2a so that it can now bind to fibrinogen. And this is rather cool, what happens next. So basically, what happens is if we draw the protein fibrinogen, and fibrinogen is a player that we're going to see again later, okay? So fibrinogen looks kind of like this. So this is how we will draw, at least in cartoon form, the protein fibrinogen. Now, fibrinogen is a component of the blood, so it's within the blood. It's a protein that's produced by the liver, and it's within the blood. It is actually a clotting factor, and its other less used name is factor 1. Okay, so it's clotting factor 1. Right, but you won't generally hear fibrinogen called factor 1, but if you're wondering what factor 1 is, there's your answer, it's fibrinogen. Okay, so basically, if we've got our platelet here, and this has its glycoprotein 2b/2a here, so this is glycoprotein 2b/2a, and this glycoprotein 2b/2a protein has been activated by the binding of thromboxane A2, so we'll draw the thromboxane A2 over here, so this is TXA2, thromboxane A2, okay. Then what this protein can do is it can bind to the fibrinogen, so it can bind to one of the lobes of fibrinogen. So fibrinogen has these two lobes with a linker in between them. Now, what do you think is going to happen? Well, if we have another platelet here that's also got a glycoprotein 2b/2a protein on its surface, which is also bound to thromboxane A2, then what can happen? The two platelets can bind together, and this is what's known as platelet aggregation, okay? Well, just two platelets binding isn't quite what we mean by platelet aggregation, so let me just continue the story a little further. So, do you think that each platelet just has one single glycoprotein 2b/2a protein on its surface? Well, of course, the answer is no. These platelets will have many glycoprotein 2b/2a proteins on their surface. So to get the point across, I think drawing 4 of them is enough to get the point across. So, I won't put one there because it's already got that thromboxane A2 label sticking there. Now, in reality, these will have absolutely loads of these proteins bound to them. Okay, so I'm drawing a few more here. Okay, and all of these may well have thromboxane A2 bound to them. So these are all glycoprotein 2b/2a proteins. Okay, so now let's have the thromboxane A2 bound. So here's a thromboxane A2, because remember, we've had this chain reaction of platelet activation. So we've had absolutely loads of platelets dumping thromboxane A2 into the blood, so it's likely that they've all got thromboxane A2 bound to them. So let's have thromboxane A2 in red here. Okay, so in red, this is, ooh, it's really horrible, the dirt on these highlighters. Not pleasant. I really need to clean these. It's ruining the pictures. It's making them all sort of dark and gloomy. Right, okay. So, green's the only sort of clean colour anymore. So, here is another platelet then. Okay, and here's another platelet. And, of course, these GP2B/2As, once they've bound to thromboxane A2, they can also grab another fibrinogen molecule, and you can see what's going to start happening now. You're going to have these platelets also with glycoprotein 2B/2A bound to them, and with thromboxane A2 bound to the GP2B/2A. Okay, and you're going to start getting the build-up of a massive, great lattice of platelets. And this is what's known as platelet aggregation. When you get platelets sticking together in this way via binding to the binding to a lobe of fibrinogen, and then the other lobe of fibrinogen binding to another platelet. This is what's known as platelet aggregation. So let's now put the whole story together. Let's put platelet adhesion, platelet activation, and platelet aggregation together to get the formation of what's known as the primary hemostatic plug. Okay, so let's draw our picture again. So here's the basement membrane here. Okay, like so. And here are our endothelial cells, which have been, well, here is our endothelial cell as far as this picture is concerned, which has been cut in half by whatever made the wound. Okay, and you've now got von Willebrand factor exposed to the components of the blood. In specifically, we're worried about the platelets. Okay, so what colour did I show von Willebrand factor in? I think I'll do it in vivid purple because it's a reasonably clean highlighter. Okay, but it still looks ridiculously dark. Okay, maybe it's just the light. Okay, so here's our von Willebrand factor, and I don't know why I've drawn two of them. I'm only planning to use one. Okay, so here comes our platelet here, and then we've got our GP1B95 here in blue on our platelet. Okay, so let's label these all up. So this is GP1B95. Okay, so 9,5. Okay, and this here, this is von Willebrand factor. Now, what will happen is other platelets will go in through this crack. So, roll back a bit. Okay, so you've had this platelet come, bind to the von Willebrand factor via its GP1B95, and this is platelet adhesion. So some platelets would here around the opening of the wound, so around these exposed and damaged endothelial cells. Okay, whereas other platelets will charge on through into other realms. Okay, so they'll go maybe and touch the basement membrane, they'll find the subendothelial space, and they'll be exposed to collagen. So they will become activated, and they'll start setting off this chain reaction, which we know leads to very high levels of thromboxane A2. And we know that this thromboxane A2 has two functions then. Okay, it can cause vasoconstriction. So one, that's a very important part of hemostasis actually. So I want to stress it. So it causes vasoconstriction. And now what it's going to activate is platelet aggregation. Okay, so the other thing I want to discuss is the fact that it's not necessarily this platelet that has adhered to the von Willebrand factor that's exposed on these damaged endothelial cells that is undergoing the activation process. There may be others that have gone in here which are undergoing the activation process. And all that activation process leads to is this higher thromboxane A2 level. Now, we know what thromboxane A2 is going to also do. It's going to bind to our other protein that's on the surface of this platelet here. And we'll have this protein in, oh, it was in green actually, let's have it in green, which is GP2B/2A. Okay, so here comes thromboxane A2, and it's now bound to GP2B/2A. GP, oh dear, we're not going to be able to fit this in here, 2B/2A. Okay, there we go. Right, so now what can happen is this GP2B/2A can then bind to fibrinogen here, one of the lobes of fibrinogen, and then this other lobe of fibrinogen will then bind to an activated GP2B/2A on another platelet, and you're going to start getting the assembly of a platelet aggregate, one of these things that I've drawn here. You're going to get that starting to form here. So what you're going to get is platelets all aggregating into a jelly, sort of viscous mush, basically, in this gap between the two endothelial cells, and this is what is known as the primary platelet plug, or the primary hemostatic plug. So you'll sometimes hear people refer to this as the primary platelet plug, but it's more properly now called the primary hemostatic plug. Now, you'll also, a very common thing that people call this is they'll just call it a primary blood clot. Now, that is not really correct. Blood clots really, it's not a very medical thing to actually say. People shouldn't use that. People do because it's very, very pervasive in non-scientific literature. So, you know, people talk about, I'm on this drug to stop my blood clotting, but it's not clear whether when people talk about blood clotting, whether they're talking about hemostasis or whether they're talking about thrombosis, i.e. the formation of this physiologically, i.e. hemostasis, or the formation of this pathologically, i.e. thrombosis. So people try to get you to avoid using the terminology blood clot. But it's so pervasive in society that you will hear people refer to this as a primary blood clot. So it's a primary hemostatic plug is its proper name. Okay, so in essence what you've got is platelets piling into a massive great mass here, and that mass, that viscous jelly mass, is what's going to block this gap that is allowing blood to leave the lumen of the blood vessel. Okay, so now what we're going to move on to is how to make this primary hemostatic plug stronger, basically. And we can make it stronger by adding an extra component to it. So at the moment it's just got in platelets, basically, and we want to make it stronger. So the way we can do this is by intertwining the platelets with a mesh. So basically at the moment what we've got is platelets like so. So we've just got loads and loads of platelets aggregated together into a great mush, okay? And this is the primary hemostatic plug, or the primary platelet plug. What we're going to now do is we're going to have these platelets again, but then they're intertwined between them. What you're going to have is a huge protein meshwork, basically. So let me show this. So you're going to have a huge protein meshwork sort of intertwined in between them, and this is going to make this stronger. And this protein that you're going to have intertwined between the platelets is going to be fibrin strands. Okay, so we will discuss how you are going to produce these fibrin strands from components which are again within the blood in the next video.